Hello. Okay. Sorry. I think my voice was not. Is my voice is now audible? Am I audible now? Okay, fine. So yeah. So today, I'm Shobik. Sorry, I forgot to turn on my mic. So I'm Shobik, and I'm doing integrated PhD in biological science in Indian Institute of Science, Bangalore. And so today's session is there. So if you have any doubt from previous sessions or any sessions, you can always mail me in this mail ID. And so if we start, so yeah, so everything from whatever I have included in this class has been taken from either Leninger or Watson or Weaver. So for this portion, I would always suggest please go through this. Always go through Watson Molecular Biology. If you don't have the ebook, I have given the ebook here, and because this is the kind of the book of the molecular biology, and you can whatever has been taught in the class, you will get here in this particular book. So if I start today, yeah. So anything you want to, okay, from previous class, if you have anything doubt, you can please go ahead and ask. I'll wait for like some, if you have any doubt. Okay. Uh, okay, then if you don't have any doubt, then we can go ahead and start the session. Okay. Yeah. Don't have a doubt. Fine. So, is the slides visible? Okay, see. Okay. I think here it is not visible. I'll share my screen itself so that you can visualize things. Yeah. This is much better. Okay. So first thing, okay. So there was, so I'll just give you a brief of whatever has been taught in this class, and then we'll solve some questions. That's what we generally do, right? And yeah. So first thing is that, so yeah. So when first DNA replication was actually someone observed. So first question was, okay, we have DNA replication. Now there are three ways we can synthesize strands, right? Or you can replicate the DNA. There was semi-conservative, where we have this like from two mother strands, one parental strand is conserved in the two copies. We have conservatives where we'll synthesize two exactly new strand come uh, from the mother parental strand, which is the blue ones, correct? Or dispersive, where we have a mixture of parental uh, as well as the new daughter strands, correct? Now, to solve this, Messelson and Stahl will uh, Messelson and Stahl, yeah. So they have got Nobel. So Messelson and Stahl, they have actually uh, got the so they have actually designed the experiments, correct? So this is the experiment. So what they've done is they have used high density cesium chloride, which will have high density, so can be differentiated in the high density N15 level E. coli, which is have a cesium chloride gradient during the ultracentrifugation, correct? So what they have done, so they have first cultured E. coli in the normal cells, then they shifted that. N15 level cells to N14 level cells. Now, if we see the different types of these bands from N15 to N16, so now let's see that whatever is possible. Okay, so N15 has higher molecular weight, so HH. So first they have cultured in N15, which have a higher molecular weight, and then they shifted it to the lower molecular weight, correct? So now, if it is a conservative reaction, so if we from 1 HH, so 2 LL. Now, so you should get a equal quantity or equal intensity of bands of low as well as H, because from HH, the conservative, from HH we are getting 2 HH as well as 2 LL, correct? So after first generation, if you see that then but if you see in the first generation, which is this one, they did not observe this kind of two specific bands, correct? If you see this particular band, they observed only one band. So that means semi-conservative, so that means semi-conservative replication is not happening, correct? Sorry, conservative replication is not happening. So at the first generation, for both, if it is semi-conservative, then also we'll get a HL mixed band, right? Here also HL mixed band, here also HL mixed band. You can see. So now, if it is conservative, sorry, if it is dispersive or semi-conservative, now how will you differentiate that? Now you grow the DNA for another generation. If you grow the another generation, you can see in this case HL. If it is semi-conservative, then it will be HL, LL, or and this from LL and LH, correct? Because one H strand will give one HL and while L strand will give another LL. So semi-conservative because one mother strand is conserved. So what we see in the gradient, that either LL and HL band, correct, at the two generations. Or if it is dispersive, then what will happen? So you will get a mixed band, right? After this one, you will get a mixed band. So HL mixed band one. Now, if you see here, again in this, if in dispersive, there will be 25% H band, but 55% L band. But in this case, you will get 50-50%, right? 50% will be LL and 50% will be HL band, correct? Now, if you see at this position here at 2, this position like 1.92, you can clearly see there is two bands, correct? So that in case of mix, there will be only one band of like H and L mixed, correct? Because there is no separate strand. So there is no strand separation. All the strands have some mixture of H and L. So that you should see only one band. But however, at 2.2 as a 1.9 or to second generation, you can see there is two bands. So this proved that no, DNA replication is actually semi-conservative. So where the one of the mother strand give rise to the other daughter strand. This is a genius experiment, and yeah, so this proves that DNA conservation was semi-conservative, correct? Now we'll move forward to okay, next slide. So okay, so the basics of DNA replication. Now DNA replication is a strand synthesis, right? So you have a template strand, and on template strand there is a primer. On that primer, with help to DNA polymerase, this NTPs comes, and from this NTPs, this catalyzation happened where it gives rise to one pyrophosphate, and which gives rise to the strand action. This is a cyclic reaction, but H reaction this goes, and it's important to remember one thing, that one of the enzymes known as one of these enzymes always use divalent cation Mg+2. All of this enzymes, okay, use a divalent cation Mg+2. We should remember that, okay. So next, so what they have done is here. Now there was a question that, okay, now this is a base pairing. Now how does this catalysis happen? From where this energy of this catalysis occurs. So if I have just did a excerpt from WhatsApp, you can clearly read it. So what happens when you have like normal reaction, what is happens that during catalysis is OH3' OH group of the previous nucleotide will attack to the coming NTPs, and from that XMP to XTP plus XMPN, this is the XMPN plus XTP, it will give rise to the access to the chat. So then the XTP and XMPN will give rise to XMPundal PP. Now this has a free energy only of like minus 3.5 kilocalorie per mole, which is very less compared to this. So how this reaction is then driven at the forward reaction. So this answers from another enzyme. There is a pyrophosphatase which cleaves this diphosphate bond and give rise to 2-pyrophosphate, 2 inorganic pyrophosphate, which has a minus 7 kilocalorie per mole. It's a double the free energy, and the equilibrium constant is also very high, which is like 10 to the power 5. So that means DNA synthesis reaction is effectively irreversible, as this says, right? So XTP, so the DNA replication reaction is XTP, which is the coming NTP, plus XMP, which are monophosphates were there, into N, which is like a DNA XMP into N, correct? DNA is monophosphates, but polymer of a monophosphates, correct? Plus XMP, so therefore they condense, and from 1 XMP N plus 1, the strand is increased in size by N plus 1, and 2 pyrophosphates. Okay. Okay. So in E. coli there are 3 DNA polymerase. In E. coli there are 3 DNA polymerase, namely Pol1, Pol2, and Pol3. So Pol1 was the first identified the polymerase which has been also renowned by the scientists, and then so this E. coli polymerase 1, it has 2 activities. So it has one, we'll write is here. So polymerase 1 has 2 kind of exons. In addition to the polymer, that is 5' to 3' exonuclease activity, and 3' to 5' exonuclease activity. Okay. So this, what does this 3' to 5' exonuclease? This has been demonstrated here. So what it is demonstrated here is that, so when there is a parental strand, there is a primer, right? Here is the primer. So here in the lagging strand there will be one primers, correct? Now this primers is a RNA primer. So this RNA primer needs to be removed, correct? Because during DNA synthesis, as in the last slide is told, there will be a RNA primer. From there the catalysis starts, correct? So DNA RNA primer. So how these primers are actually removed? Because these primers are RNA primers, you should not have it in the DNA. Now what happens? This DNA polymerase 1, it has something known as nick translation activity. What does it does? So first, this is known as nick translation. This whole process is known. What it does, as you can see, there is a nick because RNA and DNA cannot form a joint here, and there is some RNA fragment. So DNA polymerase 1 binds here. It cleaves off all the primers. It degrades the primer here and synthesizes. There is a 5' to 3', correct? So it's synthesizing at the 5' to 3', as well as using this 3', 5' exonuclease is cleaving the RNA primers. Okay, and using this RNA, by cleaving this RNA primers, what it does is then their DNA ligase will come and there the nick will be repaired, correct? Ligase will join the nick. So as you can see that this 3', 5' exonuclease activity is very important in E. coli polymerase 1 for this nick translation. Okay. So there are several things which you can actually, if you want to like interested in experiments, so then you can go to read this another book known as Weaver. This has very good like experiment how actually was DNA replication was actually different structures of DNA replication, how is actually solved the first chase experiment all and zero. But this cannot be covered in this course because DNA. So also this DNA polymerase one was discovered by Arthur Kornberg, who got the Nobel Prize in uh, 1959, and which was and shared the Nobel Prize with Ochoa, who's actually discovered the RNA polymers. So also this, that's why it's also known as the polymers one is also known as Kornberg polymers. Okay. Anyway, so this is the leading strand and lagging strand synthesis, as you can see. Now so DNA polymer. So now the most of the DNA replication is done in the bacteria is the Pol3. So how Pol3 was actually, how Pol3 was invented. So what people have done was in E. coli, people have people have like used some means to degrade or to knock down or inhibit the polymerase 1 activity. But they were seeing that okay, cells were perfectly viable. There were no issue in the cells. So how that has been done? Then they understood really okay, there should be another more enzyme that actually is having the role of this use as the role of the replication. So then they found out that DNA polymerase 3, which is the uh, primary polymerizing enzyme in this. So what they have done, they had like made mutated strains which lacked the pol1, but still the cells were perfectly viable. So that's how they found out the DNA pol3, and DNA pol3 is synthesized by DNA E gene, whereas the pol1 is synthesized by DNA B genes. Okay. So there are several subunits of this DNA polymerase, namely as alpha subunit, and the main core or the polymerase subunit or the hollow enzyme, what is called, which has caused the main subunit, is made up of alpha, sigma, and is a dimer of alpha, sigma, and theta. As you can see here, this is the core subunit. Then there is this gamma subunit, what is the DNA dependent DNA ATPase, which has this uh, kind of like sliding clamp activity, and these have this ATP binding and the other SSB binding protein activity, which is known as the Pol3 star, which is like Pol3 subunit minus the beta clamp. The beta subunit is very interesting. It's is used as sliding clamp. So DNA, as you can think of a DNA synthesis, if you can think of that, so there is a DNA synthesis happening, correct? So now here is the pol1, and it is synthesizing the DNA, okay, at this position. Yeah. So let's choose some other color. Yeah. So it is synthesizing the DNA in this direction, say. Okay. Now after synthesizing this position, this much position, if this is my enzyme, it falls off. Okay. So it falls off. Now to rebound to the strand, they at that position the sliding clamps or this part helps. So what sliding clamp actually helps is that to, if the DNA once falls off, the sliding clamp will take the DNA and take the enzyme again and to bind it to the next part of the synthesis. So if you delete the side sliding clamp, the processivity of the DNA replication goes very low. So there are two terms I think you should also know, as is known as, one is known as fidelity. One is known as fidelity and one is known as processivity. There are two terms. Okay, this was very important term whenever you are demonstrating any polymerase enzyme. So fidelity means that low error. Okay, so DNA polymerase 1 have a error rate of 10 to the power minus 4 to 10 to the power minus 6. Okay, around that. Okay, that means that it will have only one error, because DNA polymerase cannot have any error, right? These are polymerases, so they very important for maintaining the genome. So they have and around one error in 10 to the power of 4 to 10 to the power 6 base pair. Okay, and if you combine actually with the DNA repair enzymes, actually this goes down to 10 to the power minus 8 to 10 to the power minus 10, actually the error rate. So as you can see, the replication the error rate of DNA replication is very low, because errors in DNA can lead to mutation which can cause havoc, right? Because it can lead to extinction of that particular species, right? Then another thing is known as the processivity. Okay, the processivity. What is processivity? Is the number of base pairs number of base pairs Paul can do in one go. Okay, before falling off in one go. Okay, and I'm not sure that the exact number of the processivity of the DNA polymerases. Yeah, I'm not sure the exact number. I am not able to remember now, but I will include in the slide. I'll see and include in the slide. So, but all the DNA polymerases highly processive enzymes, so they can synthesize a good number of strands before they fall off and they then regain. So basically this particular thing is known as processivity. Length of this particular thing at one go, how much part it can synthesize, is known as processivity. Okay. So as DNA, so in brief, the DNA replication initiation. So DNA replication, as you know, there is a origin of replication, and there are several factors in original replications, and which are mainly AT rich factors. And then here the DNA A protein and protein binding sites, and this is the Warai site. So I am not going to details of this whatever, because it has been already taught. And if you want to go this, if you got again, you can go read Watson. These are very well explained in Watson. And if you want to do it in a short way, then Relinger is also a good book for this kind of thing. For prokaryotic replication, I think Relinger will be a good enough book. But for transcription translation, please read Watson. So you will have a very good conceptual idea how this molecular biology thing goes, because from this part, lots of questions from this week 6 part, lots of questions come in your competitive exams. So like JAM or GATE or NET. So do read of this particular portions very well. Okay, and if you have any doubt on this particular parts, please send me a email, because this part is a very important part for your any kind of competitive exams. Okay. Okay, so what happens in the in E. coli, the origin is known as ORIC. So at the ORIC, there is a origin binding proteins are there, and this DNA ATP, so they bind. Then the After so DNA ATP is ATP binds, it's the denaturation, and the DNAB or the DNAB helicase, it binds to the this particular origin of replication, and the SSB single strand binding proteins, they are also bind around here, and from here the DNA is melted and the DNA replication starts from the origin. And as you can see, the DNA replication is a bidirectional. Hello? Yeah, please tell. DNA replication is bidirectional. Okay. Yeah. So this kind of bidirectional fog, there is some experiments known as pulse chase experiment. Okay. Pulse chase experiment, and this is again is a very interesting experiment. So to prove that the DNA replication was pulse chase experiment. So prove that DNA replication was bidirectional. Now what they have done, the pulses. So now what you can do is always, since it's phosphates, so phosphates, as you can always use P32, which is a radioisotope, right? Which is a radioisotope. And I hope that I could have I should have used one. I should have include this. I can see. This is just extra things I'm teaching. Okay. So what you can do is, one is a DNA replication is going on. You can do the DNA replication outside using this clinofragment. Now what people have done, they have used this pulse chase. So wherever there is so at small pulses or few pulses of this particular P32 was given in the during the DNA replication in like some within few seconds or few months. So what if there is a one day by single directional, and if this is if this is my origin of replication or ORI, if I give the pulses, so what will happen if this is monodirectional? We should see this kind of, right? But this was not seen. What people have observed is that during the pulse chase experiment, if this is my ORI, then as you can see, people have observed like this. So that means the DNA replication is going from the origin both at direction as well as direction. So this is known as the bidirectional replication or theta model of replication. If someone tells you in some competitive direction what is theta model of replication, this is the bidirectional replication. Okay, and this was first explained by a pulse chase experiment. I don't remember the scientist name now. The scientist name, I don't remember the scientist name, but that I'll yeah. J. Huberman and A. Sai have performed this kind of autoradiography. Okay. I'll also include the I'll include the image here also. Okay. Okay. So this was all about replication. Since we don't go into very details of replication, if we go into very details, it becomes very complex, and replication will stay till the prokaryotic only. Okay. Next, we will go to the transcription. So this transcription also I'll just say. So I'm just trying to brief everything, so for like any type. So either go through the slides for any kind of like this course exam or competitive exam, just to visualize the molecular biology. So transcription always starts at a promoter sequence. So what is the promoter sequence? Two promoter sequence. Please someone this should be tell. These are very basic. Someone in the 11th law chapters. So what are the name of two promoter sequences? I want you to unmute and say. Come on. three people whoever have joined, you have to tell what are this promoter regions are called. Supardha, Samantha and Shakti. Can you please unmute and say what are this -10 base pair and the -35 it is called? No? Okay, anyway. So this is the consensus sequence. This is the up element. Up element is helps in the DNA RNA transcript, but it's not exactly necessary. So anyway, so this part is called data box because it's a signal. So because sequences are TATA or Pribnow box, the name of the scientist. Okay. And this is the this is the another -35 sequence, which TTGA. Okay. So these two are, so DNA replication always needs the promoter region, which are two consensus promoter region -35 as well as -10. And these promoter regions are very important because there would be promoter binding proteins. Okay, promoter binding proteins, or also known as transcription factors. Transcription factors. Okay, transcription factors. So these people binds to this promoter region and activate or deactivate these promoters. Okay, this is very important because during the expression of the genome or in the DNA, this kind of promoter binding proteins or transcription factors, they actually play a very important role to control the expression from the genome. And I don't think it will be taught in this course, but different nucleosome and nucleosome modifications actually bind or unbind this kind of regions. This is still a very active area of research. So how this kind of organization in the genome is, in this compact genome is mediated. So if you are interested in research, go and search this particular term is like uh, chromosome organization in cells. Search this chromosome organization and gene expression in the cells. You will get tons of paper, and it's still very active area of research. Okay. Anyway, so transcription always starts from this site. Okay. So after that, so in case of prokaryotes, there are several other things bind. So the core polymerase bind in case of this of prokaryotes like E. coli. So this polymerase is bind. It's complex, open complex, and after the transcript, it forms a closed. It binds to the closed complex. DNA at this melt the DNA at this particular positions, and then it forms the open complex. From this open complex, start adding the some nucleotides, and after at first if it starts to add 6 to 8 nucleotides, and after that when it crosses the 10 nucleotide bound, average 10 nucleotide gap, then it starts synthesizing the RNA from the protein. And as you can see, there is the RNA and protein is there. And yeah, so then transcription is terminated by using several factors, which will be coming to the next slide. In case of in case of termination, so there are two termination: row independent termination and row dependent termination. So in row independent termination, there will be a poly U sequence along with a hairpin structure. and in previous that there will be a GC region also. So during this kind of row independent transcription termination, the this kind of RNA hairpin as well as this AU. So AU hybrid is very much unstable. So that leads to the release of the RNA from the RNA DNA hybrid, and their RNA polymer is also falls off. This is row independent transcription termination. And a row dependent transcription termination, there is a row helicase which binds to the RUT site known as row utilization site of this particular type of mRNA which have this RUT site. So it binds. Now so row is a DNA helicase, sorry, RNA helicase, which goes and runs through the RNA. Okay. Now what happens in cases where there is a hairpin as well, hairpin structure in the near the termination site, which is not has been shown here, but generally there will be a hairpin structure here, and the RNA polymerase is great slow. So now during the transcription, normal transcription when it is happening, and at the mid phase, both polymerase is also moving forward as well as the row is also moving forward. Now what happens during this forward movement? Row cannot catch RNA polymerase because the RNA polymerase moves also very fast. But when there is a hairpin or some kind of structure during the near the transcription termination size, row catches the RNA polymerase and then it removes this RNA DNA hybrid, and thus it frees the mRNA. So this kind of mRNA needs to have this row utilization site or RUT sites. Okay. In case of eukaryotes, so eukaryotes, the overall principle is very same. However, there is just some extra factors which are coming to here, as you can see. So first, there is the Pol2 is the DNA polymerase that cause the RNA synthesis from the mRNA synthesis mainly. So it has a 12 subunit. Then so first at the Tata binding protein binds at this minus 30 position at the Tata binding protein binds and recognizes the Tata box first. Okay. Then the TF2A as well as the Tata with help of Tata binding protein basically binds to the this Tata C region or promoter region with help of TF2A. Then TF2B comes and TF2B recruits the Pol2 TIF2F complex. So this is the complex. After that, so then TIFE recruits the TIFAH, which the helicase. TF2F recruits the TF2H, which is the helicase, and this whole complex melts the DNA to forms the open complex or bubble form. So as you can see, this is the kind of the how do I say the order of why it should bind. So first the Tata binding protein binds, after that the TF2B binds, then after that TF2F polymerase binds, then after that TF2E with help of TF2H with help of TF2E binds, which forms the older complex. So so this order is remember this order this is important. Okay. So after that overall the replication is really similar, and there is transcription termination as well as this C-terminal domain dependent dephosphorylation, and those things happen. So I am not going into details of this because there is a dephosphorylation and phosphorylation of the C-terminal domain of this of the Pol2, which leads to the formation of the open complex and all, but I am not going to details of that. Okay. So there are post-transcriptional modifications. So one of them is the 5' cap. As you know, the cap is very much important for stability of the RNA. There are several mRNA decay pathways. We actually removes the cap to make the RNA available for decay. Correct? Okay. So this caps actually prevents the RNA from this exonucleases, which cleaves the RNA, several RNAs. This is the structure of the RNA, which has a 7-methyl guanosine cap. So this is the pathway which has been already taught in the course, and I don't think I have to explain something. It's a very basic thing. So basically, from phosphohydrolase, it's from the PPNP. When guanyl transferates, put a guanine group on this GPPNP, and then from a adomate to from adomate, the methyl transferase put M7 in the GPP, and then this again another adomate puts another M7 here. So there's like 7-methyl guanosine, and it forms the whole cap. So I am not going to saying in detail because it's already quite easily explained, and I included this figure also. This you can also go and read in Watson. It's very well written. I have taken this image from Ledinger. Okay, then there is RNA splicing. So splicing is a self-driving reaction, molecular reaction, which is controlled by some of the binding of some proteins. So during the group 1 type of intron, there will be a UPA and there will be a GPU. So this which or from those, this UPA will attack to this guanine, and this will lead to cleavage of this particular side, and then this particular group, like there will be a guanosine phosphate here, which actually will attack as a nucleophile to here. This will leads to breakage of this particular phosphodiester bond, and then this particular G attacks this particular wedge and cleaves off. Okay. In case of group 2, so there is a, as you can see, there is a external guanosine which acts as a nucleophile. In case of group 2 intron, there is no external guanosine phosphates is guanosine phosphates is like acting. Instead of this, you have this kind of sequence CPA-PA. From there, the wedge from this A attacks the G here, which in turn attacks the PU here and forms like a lariat-like of structure. So this kind of lariats is also known as circRNA, which is again also a quite active field of research. Okay, this is also known as this lariats or lambda. So as you can see, there's in group 3 introns, again I will not go to very detailed. So there are overall the idea is same, but then there will be several splicing proteins which will actually bind to the spliceosome, and you can see that there is a spliceosome which is being activated by phosphorylation, and there is several things, but overall lariat formation and everything is same. Okay. Instead, in group 3, the speciality is that it's much more controlled, and because it involves drop sites of this splicing proteins. Okay. And I'm not going into details. Then there is a polyadenylation also. As you know that polyaddition is also very important because it increases the ribosomes. Increases the ribosomes efficiency to bind to, not to bind, like to increase the efficiency of translation by recycling the ribosome in a well defined manner. Okay. And yeah, so what happens during the transcription, there are polyase signal sequences in the RNA, also known as PAS. Also known as PAS. PAS or polyadenylation signal sequence. So this type of signal sequence, this type of signal sequence is there. There will be two proteins. CSTF and CPSF will bind to this kind of proteins, and it basically binds to the RNA polymerase also. These proteins jump onto this polymer signal sequences. This RNA cleavage is done by CPSF. Then polymerase comes to create the CSTF. So polymerase bind, and CPSF and CSTF cuts the RNA from here. And polymerase, which is the template independent RNA polymerase, it creates some polyA tails, around like 200 polyA tails, and then the polyA binding proteins bind to stabilize this polyA tails. Okay, and this polyA binding proteins or PABPs are again very important proteins. These green things are PABPs or polyA binding proteins. So PABPs. So these are again quite important to maintain the stability of the mRNAs, as well as several other functions of the mRNAs are controlled by this polyA binding proteins. Again, this is also still now very interesting area of research, and you can also go and have a look at how PABPs. There will be several review papers. Okay, so now we'll move on to the last part of the central dogma, that is the translation. So why it is known as translation? Okay, can someone tell why it is known as translation? Why something not other term? Okay. Translation means, because it's known as, because the DNA or RNA languages, DNA or RNA language, there are mainly 4 nucleic acid or four letters, right? In RNA language there will be A or T or U, G and C. Now this is a DNA RNA language, but protein, as you know, the languages is a little different. So it has 20 letters, correct? It has 20 letters because it has 20 amino acids. So I am converting a 4-letter information to a 20-letter info, correct? So like in translation, we convert English to Hindi or English to Bengali or Bengali to English or English to German, several other languages. Again, I am converting the DNA or RNA language to a protein language. So that's why it is known as translation. Okay, so what is this language? So how does this language works? So 3 DNA or RNA letters is equals to 1 protein letter, correct? 1 protein letter. Okay, so when I am seeing a stretch of DNA, say T, G, G, C, G, C, C, G, T, G, like that. So this will correspond to 1 amino acid, this will correspond to 1 amino acid, this is corresponds to 1 amino acid. So if it's a 9 nucleotide sequence, so if it is a N letter coding sequence, I should get n by 3 minus 1 letter protein sequence. Why it is minus 3? Why there is a minus 1? Why there is minus 1 I am putting? Why there is a minus 1 I am putting suddenly? I told. So stop codons, correct? So there are start codons as well as stop codons. What are the start codons? ATG, correct? Mainly, canonically, ATG, sometimes GTG also. What are these stop codons? TAA, TAG, TGA. So if there is a TAG, I will have some from ATG. I will get something. Say I will say X. Where is the methionine? Then I will say Y. Then there is Z. But if there is a TAG, I will not get anything. Translation will get terminated. Correct? So from this, so this 1, 2, 3, 4. So what 3, 12. So on this 12, I am getting 12 by 3 minus 1 is equal to 3 amino acid peptides, correct? That's why it is n by 3 minus 1. This minus 1 to denote the stop codon. Because stop codon doesn't give rise to any amino acid, it terminates the translation. So for translation, what do you need? We need the tRNAs. Now there will be several tRNAs which will have this proper codon. So all these triplets, which triplet is known as codons, and codons binds to the codons are in mRNA, correct? What are their codons are in mRNA? What there is in tRNA? So in tRNA we have anticodons. Now there is a very interesting thing I'll tell you. I don't know if it is written here properly, but in codons are in, when codons are written, it is written as from 5' to 3' direction. The anticodons, if I have ATG, the anticodon will be for the ATG. So if this my ATG is my codon, the anticodon will be G. So C T A T. Codons are written as from 3' to 5'. This is very important thing you should remember. If someone says you but, okay, but normally when we write a sequence, we write always a sequence in 5' to 3' direction. So someone asks you, okay, what is the codon for this anticodon? If I give you an anticodon, okay, if I give you an anticodon, I am just writing is ATC anticodon. If I give an anticodon CAT, what should be the codon? So first, this is, always remember, this is 3' to 5'. So what you should always do is just convert GTA. Now this is also in 3' to 5'. Correct. Sorry, I by miss. Sorry, my bad. Oops, sorry, my bad. So remember, this is written as. So always we write any sequences. Always we write at 5 prime. So this is the 5 prime. This is the 3 prime. Now whenever you write, remember at the codon it is on the opposite side. So this is 3 prime. This is 5 prime. If you write this, correct answer. Okay. If you just write this, it's a wrong answer. If you just write this, correct answer, because by convention we always write any sequence from 5 prime to 3 prime orientation. So even if you don't mention, okay, this is my 3 prime, this is my 5 prime, I will say that okay, this is correct because he by convention he has right. But if you tell me this without mentioning this is wrong, because I'll then think the sequence is GTA, which is wrong. Always remember this particular thing. The codons in the mRNA are always written at the 5 prime to 3 prime, whereas when the tRNA sits basically on the codon, it basically sits like if this is its 5 prime and this is its I should write the proper way. So I should write, yeah. So if it is my DNA and if this is my mRNA and if this is my codon, always remember this is 5 prime to 3 prime, but when the anticodon binds here, so if I just write so no, this is kind of the mRNA structure, the tRNA structure, right? Right, this is the tRNA structure, correct? If this is my tRNA structure, always remember that the tRNA is, if this is 5 prime RNA, the 5 prime of the tRNA is this. This is the 3 prime of the tRNA. CCA. 3 prime CCA. Okay. Always remember this. This is a very important concept you should always remember. So this is on from 5. So this is actually the anticodon sequence. If you just flip the anticodon from ATG to just TSC, it means that this is 3 prime to 5 prime. Just opposite it. Okay. So do remember this. Okay. So yeah. So then there is also what I was just telling is that there is a charging of tRNA. So all the tRNAs are charged with amino acids. So based on the whatever anticodon it has in this position, it gets charged to an amino acid. So first the amino acid tRNA is formed by using the amino acid and with the ATP. Using that ATP energy, first the amino acids, use the ATP acid, that adenylated amino acid is formed. This adenylated amino acid then is attacked by the 3' OH group of the CAA. Here I have also written CAA. You should go and read to the tRNA structures. Everything I can cover, but you should always go and write. And this kind of CCA, sorry, not CA. CCA is important because the 3' OH of this CCA attacks to this adenyl here in this particular bond, and AMP is released and gRNA is charged. So this is a very high energy bond and it can be easily broken during. So this amino acids get incorporated when there is a peptide bond formation. Okay. Okay. So the translation initiation in prokaryotes. So prokaryotes have this initiator tRNA known as N-formyl methionine or FMET. So this initiated tRNA goes and binds to the, with help of IF2 and IF1 and GTP, it binds to the A side, and then this initiated tRNA goes and forms the 30s p initiation complex, and finally with some other proteins it forms the 70s initiation complex. In case of eukaryotes, it's a little different. So the in case of here you can see first the ribosomes subunits bind, after that, and then first the small subunit binds to the mRNA, and then the large subunit comes and binds. Okay, in case of uh, eukaryotes, there are several other initiation factors which binds to the cap as well as in the mRNA, and there are other factors which binds to the small subunit of the ribosomes. So when the small subunit of the ribosomes is bind to the EIF3 actually binds first to the small subunits, and to remove the large subunit as well as EIF1, and all this factor binds. Then EIF2 GTP will come with the normal methionine. So remember, there is no methionine cap here. Now this whole thing is forms from the 43S pre-initiation complex. This pre-initiation complex goes and binds to this mRNA. Then this particular from the cap binding through the cap, there is the cap and there are cap binding proteins. Now through this cap binding proteins, now it scans to the so this is the start codon. It scans to the mRNA to find the start codon. As it scans, it uses the ATP hydrolysis and forms a loop kind of structure. So when it finds the ATG, the 60th subunit comes and binds to the protein. Then several other factors put, and the elongation happens. So during the elongation, more or less it is same. The basically EFTU binds to the amino acyl tRNA and brings it to the amino acyl site, and then there is a peptide bond formation, and the EFTO reagent. Then there is a attack from the peptide bond, and the peptide bond gets formed. Okay, so this is the prokaryotic as well as the eukaryotic. So eukaryotic, you don't have this. It directly goes and binds. Here is a protein known as EFTO which use ATP hydrolysis, which is much more controlled. So translation terminations. The translation termination is done by proteins known as release factors. So release factors terminate translation in response to the stop codon. If it is found a stop codon, any of the three TAG, TA, and TAA, then then the ribosome RF factor are recognized by the protein known as release factors, and there are two classes of release factors. Class 1, which recognizes stop codons and triggers the hydrolysis, and prokaryotes have two classes of release factor, RF1 and RF2. RF1 recognizes the UAG, RF2 recognizes UGA, TAG or TGA, and third stop codon US recognizes both RF1 and RF2. So eukaryotic cells, they have only single class known as ERF1. ERF1 recognizes all kind of class stop codons. Then there is class 2 delete factor ERF3. ERF3 binds, and as you can see the ERF1 binds here, recognizes the stop codon. Then ERF3 binds and do a GTP hydrolysis to release the the RF3 binds and release the peptide bonds. Then RF3 also hydrolyzes this GTG do a GTP hydrolysis to remove the RF3 also. You can see that is the GTP binding and hydrolysis. This again I have taken from Watson. Please have a read on that particular thing. Okay, so then there are several antibiotics which can target the different stages of translation. So in case of tetracycline, it binds to the A site, near A site, so you can go and have a look there. So if you want to remember, so this particular, some of them are very important in the research purpose. Cyclohexamide, which many people they talk. Puromycin is another very important antibiotic. It prevents the peptides at the center. It mimics the tRNA. Okay. Cyclohexamide again is immediate the peptide activity. So this you should remember. Others, it's okay to remember, or remember also some of like chloramine phenicol you should remember. Tetracycline also very important. So there are several antibiotics you should remember and had how it works. Okay. Yeah. So okay. So here is the gist of our today's courses finished, and now we'll move on to uh, the assignment section. Correct. Okay. Let's go. The concept of central lemma of molecular biology was explained to Florida. Of course, it was written by Francis Crick. So yeah. So as sir told, that is, he wrote a letter to nature. What I'm doing? Sorry. Francis Crick is the correct answer. Correct. Which is the polymerase in Primus carried out? It's a DNA polymerase alpha. So you can see, if you have seen there is a list of genes which you can see that what it does in different types of proteins. Correct. So what I will do, one small thing I will do here, I will put use the sniping tool and put a table so that it will be remembered. It will be easy for you to remember. I'm not sure it is not happening. Anyway, I'll put a table for you to remember this thing. Okay. The strand formed after joining the Okazaki fragments, the DNA. So DNA has two strands, right? When there is a trans replication is happening. So in 3' to 5' strand, it is easy to happen because always the DNA synthesis is going on the single direction. But in the 5' to 3' strand, since DNA synthesis always happens on the 5' to 3' orientation, sorry. So since DNA synthesis is, I have given it wrong direction actually. Direction I put wrong. So yeah, when a DNA synthesis is always happening in this is the 3' to 5' strand. Synthesis happens in a this regular way, but in this case, it always has to always happen in 5' to 3'. So it happens like this. So these are the Okazaki fragments, right? So there is the leading strand, the lagging strand. So Okazaki fragment is from the lagging strand. Do anyone know why this is known as lagging strand? Why this is leading or lagging? It's just leading means someone is leading and someone is lagging, right? I don't think anyone knows this. I should share one small thing to you guys. Okay, let's see. I'll find it in Weaver, I think. Okay, this is the okay. I hope you can see. I should have included this. Suddenly I remembered. DNA damage repair. No. It is a beautiful picture actually to explain why it is lagging stand and what is a leading stand. I'm not able to find that particular thing. lagging strand. Yeah, I will tell you, this is a very interesting concept. Why this leading strand and lagging strand? I think I should also find it in this one also. Let's see, replication of DNA, and if I go here, yeah, this is the one I was searching for. Okay, why it is called lagging strand? So if you think of the DNA replication, so if you can visualize this one, you can think that, so one strand is going, this is a 5 prime 3 prime strand. Okay, this is going properly, correct? Sorry, this is the 5 prime 3 prime 3 prime. So this is the 5 prime 3 prime 3 prime 5 prime parental strand. This gray colored one, I should, this particular strand is glowing, correct? The sliding clamp and all, whatever structure you forget. But if it is growing, and this part, if it has to synthesize the fragment, what it has to do is, it has to first form a loop like structure here. Then only it can, because this is synthesizing in this direction, but this one has to synthesize in this direction. So how will it will do that? So first it will make a loop, okay? Then the second lagging strand DNA polymerase binds here, and then it synthesizes again this part here, A, this B. So then it's the loop is again released, A, B, C. So yeah, so primase comes here, RNA primer is synthesized. So now then second lagging strand is synthesized. So you can see primase is released. Now second lagging strand binds here, and then it will release this particular strand. I'll also want to show you one video which I have seen long back. DNA lagging strand replication. Yeah, so this one. Please see this video. Okay. I think if you can not hear the video, I should also include computer sound.
A sequence of 4 chemical bases represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3'. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous.
So this is not this is continuous. So this is a leading strand. This go fast. Now, but this strand already this part is synthesized, but this part is not synthesized yet. So it is a lagging strand. So you can see there is a looping.
Because it runs in the opposite direction. The DNA polymerase therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer.
RNA primer is getting added.
DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes the polymerase. DNA ligase sealed because each DNA and one new one.
Actually, this was not the thing I was. application. Here. Correct. This is
Like these prokaryotic E. coli cells replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell
I'm
division
not come
occurs.
just.
In order to understand how
Yeah.
the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex
Exactly.
organisms replicate DNA,
As you can see, this part is already this is the leading strand, this is the lagging strand, correct? As you can see, there is no looping here because it's already. Now this part has to be looped, correct? Because then this has to move here. This particular arm has to, I think you can see, right? Yeah, this particular arm has to move here from here to synthesize this kind of the lagging strand. Since this strand is synthesized later from this strand, that's why it's known as lagging strand. As you can see in this,
scientists first studied replication in prokaryotic models like E. coli.
see it's looping again.
A number of
See,
enzymes are needed for replication to proceed
again
once
it's
the
looping
replication
and
fork
lagging
is established.
because it's
Helicase
looping.
separates the strands of the double
So
helix.
this kind of looping only I was saying,
Replication to
okay.
proceed once the
So
replication
that's why
fork
this strand to see this particular part is synthesized. This particular part and this particular part should be complementary to each other. But this particular part is synthesized earlier, but this particular part is synthesized quite later. So that's why this is known as lagging strand. Okay. And yeah, you like virus and all.
the addition beta D have to be paired to the template strands by the enzymes of RNA called primers have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous.
So you can see this is looping out.
Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand.
Yeah.
New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand.
See this arm moved here.
Polymerase 3 then
Again
adds
it's
sequence of
lagging it out.
nucleotides,
Okay.
the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase
As I have told you the Nick
RNA
translation
primers with DNA nucleotides.
and like is coming. So
And
this is the
DNA
Nick translation
ligase
part.
is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, two identical copies of the DNA molecule result.
Okay, so this was one thing I was just wanted to show you. Okay, that's why it's called as lagging strand. Okay, to prevent the further migration of the replication frog and release the cold polymerization in process during DNA replication in Escherichia coli. So as you know, Escherichia coli is a rolling circular model replication. So there during the termination there will be this star sequence. If when the replication fork comes here and as well as this particular fork from this side comes here, is a fork here. There is a fork here. The star proteins is binds to the TOS proteins, which is helps in the replication termination. Oh, I'm not okay. Sorry, I'm not. I was not. So this start proteins is there and 2 proteins bind here. Okay. Okay. Nucleosome like structure that binds 30 merge sequence is DNA and IHF HU binding protein, DNA and HU protein binding. Yeah, 30 merge. It is formed by, I don't know, this has not been taught, but it is DNA and HU. The direction of synthesis of phosphodiester bonds during DNA replication is 3' to 5', 5' to 3', both 3' and no directionality. Remember that this strand, whenever we synthesize 3', 5', 5', 3', correct? When DNA is synthesizing at this position, there is a 3' OH, correct? When the next NTP comes, so NTP comes, it has a 5' PO4, correct? So then this particular 3' OH attacks this particular phosphate here. So that's why the directionality of this is like from the phosphodiester bond itself. This is the phosphodiester bond, right? Is from 3' to 5'. In this a very common competitive competitive exam question. Remember this, okay? Very common competitive exam question. The segment transcribed from DNA is called transcription unit, replication unit, replicon, amplicon. So transcribed, so of course replicon is from replication, amplicon is amplification. So replicon comes from replication, amplicon comes from amplification, so PCR amplification basically. Transcription unit is come from transcription, and replication unit is again is same as kind of. The AT-rich region that undergoes localized melting during the transcriptional initiation in prokaryotes is known as Tata box, GC box, Pribno box. Okay. Tata box and Pribno box, I think same, right? Tata box and Pribno box is a no, I completely agree. Yeah, so this should both this should be true. Tata box and Pribno box. I'm not sure why it is given. This question is important. The efficiency of promoter vary with respect to the consensus sequence. Consensus sequence. So efficiency of promoter vary with respect of consensus sequence varies as consequence of efficiency of promoter vary with respect to the consensus sequence. Right? Closer the promoter sequence to the consensus sequence, because conscientious means that in most of the species whatever we see and which is highly adopted, right? This kind of series DNA replication transcription is a very basic process. They are always in high selection pressure. Whatever is conscientious, it's like should have the greater efficiency than the anything variation from the conscientious. Okay, the enzyme used in synthesis of 5S rRNA. Ali polymerase 1, 2, or 3. So RNA polymerase 1, tRNA 3 rRNA 2 is mRNA 3 is DNA replication. So the correct answer is RNA polymerase 3. The correct order of binding transcription factor in eukaryotes is, so first the any so RNA polymerase this TF2H binds last. So let's see, TF2D, TF2A, then TF2BF RNA polymerase. So first TIF2 Tata binding proteins bind, right? So the Tata binding proteins with help of TIF2D and TIF2A, this is the TBP binds. Then TIF2B helps in binding of, you should remember is in this way that you should remember all the functions so that you don't have to actually remember things. TIF2EF and with help of TIF2B and TIF2F binds with the RNA polymerase. After that, the TIF2E with the help of TIF2E, TIF2H binds. Then after that, that GF2J, another protein to bind the transcription. So correct answer is A. Okay. Oops. The transcription factors that bind to the minor group of the eukaryotes is the Tata binding proteins. The multifunctional transcription factor having ATPase, helicase, and kinase-like activities TF2. Which TF2? H. Always whenever it is said, remember TF2H. H is, I just remember it like this, H is equal to helicase. So that's how you bind. That's how I remember it. Okay. It's a very difficult thing to remember. The enzyme not involved in 5' capping, as I have always told you, the guanylyl transferase transfers the this guanine to this NPP caps, correct? So it's it forms the GPP cap, as I have. So if I go to some back, here. So first the first part relay, so PPNPP guanylyl transferase, its PPNP is there. So PPNP first binds with guanylyl transferase, will transfer the guanylyl phosphate to the GP. Then the guanylyl methyl 7 transferates will form the MEM7 and orthomethyl transferase will form the another O methyl. So if I go back to this one, yeah. So then NPP to GPP, so GP NPP, then from that with particular this enzyme, then from like A, from then with help of B, it will be M7 GP NPP, GPP NP or GPNP, I forget these things very. Okay, then C is not there, we is using D, we form M7 G, I think GPN GPM NPP. So ABD is used, C is not used, so this is used. Correct. So what is snRNA is having the catalytic activity cleaves phosphoramide during the splicing. Again, you have to go to the group 3 splicing and remember this enzyme. This is a very remembering question. I don't have to explain. U1 and U6 is the correct answer. The RNA acts as a template and provides codon information and incorporation amino acid into the polymer enzyme. I don't think I have to tell you this one is mRNA because mRNA is the RNA that will have this codons, this mRNA with GP cap and this. Okay. Where actually the ribosomes will bind and form the proteins. So, the divalent colored, if you remember, I have told you Mg plus 2 was all of this enzyme. Remember, Mg plus 2 is this is the D factor. Okay. The factors preventing the association of 30s and 57. Again, I have shown you. I think remember if you have remembered. So this is the EIF3 or IF3, which prevents the here IF3, as you can see, IF3 prevents the binding of the large subunit to an uncharged small subunit. The correct answer is IF3. Okay. The antibiotic that inhibits GTPase activity of RF3. Which one is this? Again, I have shown you that in that particular list, which we have to remember, this trichodermy. Thermostable in PCR reaction, DNA polymerase 1, 2, 3, they are not thermostable. Correct? So only is this PFU. So our class ends here. So still, if do you have any doubt regarding the code, regarding anything that I have taught till now? There are several things I could not cover because it's not easy, it's really difficult to cover everything which has been taught. I think sir has covered most of the parts. It is a huge field and it's really very difficult to cover. So yeah, so that's it. Okay, do you have anything else which I should tell you? Okay, and as I was saying you something those pulse chase, I'll just show you one picture where you can actually see the pulse chase experiments or this video. So yeah, I show you the pulse chase thing. DNA damage repair. Yes, as you can see in during pulse chase, we should observe origin and from two sides, not observe what from single side origin. Correct. So as you can see, it's always observed in the two sides. If it is the original radiogram, and as you can see, always it's observed in the two sides. So that means there was the theta model of bidirectional replication. What I have taught. Correct. So this case is the theta model. And anything else I can teach you here? So you can see in Weaver there are lots of experiments wise they have taught it. You can go and have a look on Weaver, but it may be difficult for many of you. So it will be difficult. So which one? So this experiment says that which one, DNA B or DNA G, which one has this helicase activity. So certain helicase acid, DNA unwinding acid. So you have a wind like double-stranded structure like this. You put the helicase. This experiments are still done in many of the labs. So they will put some of this DNA binding proteins, and if there is a unwinding, you will see that this unwinded product, small product, is here. If it is not unwinded, you can see there is the full product. Okay, substrate. So yeah, so this is where I will end the class. So thank you everyone for joining. I'll be more happy if more people joins actually to the class. I see only 5 people or 4 people joining every time. So yeah. So thank you everyone. Goodbye.